Thank you for the invitation. Of course, thank you all for coming to hear about, from my perspective as a chemist, diving into the world of sustainable agriculture and all the other parts connected to it. I suppose because I am a chemist, this talk and my work all becomes sort of by way of an apology for sort of the framework that chemistry has set up for how we think about soil's role in plant growth and agriculture. Really, the sort of the punchline to this work is that soil science is not purely a chemical endeavor, as some chemists have traditionally pushed forward, but rather it's emerging as a biological endeavor, in particular a microbiological endeavor. So it's quite curious, and it's sort of a sentiment that's reflected by this saying that's attributed to a Chinese saying that man, despite all his artistic pretensions, his sophistication, and his many accomplishments, owes his existence to a 6-inch layer of topsoil and the fact that it rains. And it's quite remarkable that while having plants and having the correctly chosen food crops is an important endeavor to food production and our survival as a species, sort of the story I would like to begin to evolve and show you is that the viability of our ability to produce food is perhaps owed in a great part not to the plant itself, but to the soil and the environment and the context in which it grows and the fact that it rains. And so this is sort of where the chemists have their hands stuck into the issues of soil science, interest in agriculture and producing food at higher rates has always been a concern of human society, but particularly around the turn of the 19th century, chemists began to apply a scientific mind to how do you feed plants and how do you grow plants. And in this line, the father of inorganic chemistry, Justus von Liebig, started to push forward many ideas on how do you provide nutrients to plants. He correctly asserted that all the carbon, hydrogen, and oxygen that plants require comes from photosynthesis via the air and water. But as an inorganic chemist, he also pushed forward the notion that basically all the other elements that the plant requires, nitrogen, phosphorus, potassium, and a host of other micronutrients, have to come from the soil in sort of inorganic, non-volatile forms. In order to sort of supplement the existing pool of these nutrients in the soil, he put forward the idea of fertilizers, that you have to replenish and re-add these nutrients and these inorganic elements back to the soil, traditionally in the form of sort of the human-derived waste that we would use or that we would create. He also popularized the law of the minimum that said that the plant's growth is ultimately limited by the least available of these crucial nutrients. And that by balancing out these nutrients and supplying sufficient amounts, you could potentially grow larger and healthier plants. And so from the 19th century onward, this sort of idea that the soil is this repository of these inorganic elements, this sort of chemical sponge that absorbs and releases these nutrients, was a large driving force in how we approach agriculture from a scientifically rationalized point of view. However, as we've progressed through the 20th century and as we've begun to more closely examine the soil, it has naturally come up, or it has naturally occurred to us that potentially this purely chemical, relatively simple view of the soil is perhaps a bit naive, and rather we should start to analyze not just soil chemistry, but soil biology as well. And so in this talk, I'd like to sort of address 3 big questions and try to hopefully answer most of them. In the first part, I'll talk about what are fertilizers, what they do, and potentially why we can't be using chemical fertilizers forever. In the second sort of little bit, I'll talk about not soil chemistry, but the crucial aspect of soil biology, in particular soil microbiology. And then pulling together these ideas, I'll talk about what we should be doing for modern agriculture in the face of these issues of soil microbiology. And in this, I'll sort of talk about current technologies and bring in some of my work in order to address the issues for modern sustainable agriculture. So for this first portion, this issues of fertilizer has become quite important, particularly over the last year. Within the last year, the U.S. and more wider globally Most of these fertilizers and other agrochemicals, so pesticides and other additions to agriculture, were produced by 6 major agrochemical companies. Throughout the course of the last year, these big 6 agrochemical companies consolidated through mergers into the big 3 companies. And so Bayer and Monsanto joined forces and combined their agrochemical divisions. ChemChina and Syngenta also merged into one giant monolithic company. And Dow and DuPont also merged their agrochemical sections to consolidate the production and the distribution of these crucial agrochemicals. Now, this has spurred in a lot of concerns and discussion about the issues with surrounding the means of production, distribution, and research of a crucial aspect of agriculture in the hands of 3 companies. And this is also still set against the background of concerns of companies like Monsanto and their use of pesticides in agriculture. And so the big question now is, you know, in the face of all this sort of alarm and concern, we should perhaps first understand what does fertilizers do and at least get on the same page, at least from the chemical understanding of it. So as Justice von Liebig rightly asserted, plants get all their carbon from CO2 through natural photosynthesis from the atmosphere, but all the other nutrients, nitrogen, phosphorus namely, have to come from the soil. We have traditionally replenished the pool of nitrogen and phosphorus in the soil by returning all the waste that we produced back to these soils so that they can be reutilized by the plants. Now, for the large bulk of human history, and arguably still today, the concern has always been, how do you produce more food to feed an ever-expanding populace? What the chemists will tell you is that ammonia is a fairly simple chemical compound, as is phosphate. Why not produce these nutrients in a lab, in a factory, and simply add them exogenously, synthetically to the soils? And so under this idea, we would increase the amount of these 2 nutrients, these 2 fertilizers into the soils, grow bigger plants, fix more CO2, make more food. And indeed, for the bulk of modern history, at least since the Industrial Revolution, this is exactly what we've done. Here we have data from the FAO, at least from 1960, that shows that globally we've been increasing the amount of nitrogenous fertilizers that we've added to the soil. Phosphorus follows a similar trend where we have synthesized them in the factories or mined them from the earth and added them to our agricultural fields. Correspondingly, the more fertilizers we use, we have seen a rise in global food production. And not just simply food production, but not simply sort of the gross food production, but the productivity of each farm. So basically the amount of food you harvest per area of land. So this is great. Just as von Liebig was right, we add more chemicals to the soil, more nutrients, bigger plants, more food. Great. However, within this, there is also the understanding and our ability to track the realization that as we've added more nutrients to the soil, the efficiency at which these nutrients are utilized has gone down dramatically. If you look in 1960, for example, for nitrogen, in 1960, for every ton of nitrogen fertilizer we've used, we got out about 200 tons of food. As we've added more nutrients to the soil, as we've fertilized more, the rate and the efficiency at which these nutrients are utilized has gone down, such that nowadays we get about 75 tons of food per ton of nitrogen that we add, and phosphorus follows the same downward trend. Now, we are still producing more food, so potentially this is fine, that we require increasingly more amounts of nitrogen and phosphorus, but a natural question is, will we ever run out? The issues and concerns of running out is not a new one. Historically, we've seen it, particularly within the US in the 1850s, with expansion to the west and growth of sort of the American Midwest agricultural lands. There created great need for additional sources of fertilizer beyond what we could normally find within the continental US. This has sort of spurred investigation or exploration throughout the Pacific, in which they found that many islands across the Pacific were laden with bird guano, so their droppings and their leavings. From a fertilizer standpoint, guano is a wonderful fertilizer. It contains a lot of nitrogen and phosphorus in reasonably the correct proportions. But there are these sort of islands just sitting out there laden with this stuff. This sort of triggered the Guano Islands Act in 1856, in which the U.S. government basically said any U.S. citizen can go to one of these islands in the Pacific and claim it for the U.S., and allowing us to mine and ship this resource back to American farms. And so this idea that supply of nitrogen, phosphorus, and fertilizers requires government intervention, and quite drastic ones that potentially infringe on the rights of people living in these Pacific islands, is certainly not a new one, and underscores the importance of dealing with these issues of scarcity. In our modern context, we still face issues of scarcity. Phosphorus as a fertilizer has been likened to be the new petroleum. Phosphorus is a fairly difficult element. It doesn't form any volatile species that will cycle through the atmosphere like carbon and nitrogen will. Phosphorus likes to exist as phosphate rock, hard, rocky minerals that don't really cycle through the environment on appreciable timescales. As such, phosphorus has largely been considered a non-renewable resource. All of the phosphate rock that is currently in the ground is all that will be there for our given lifetimes. And so the U.S. Geological Society has begun to take stock of how much of this phosphate rock is left in the earth for us to mine and use for fertilizers. Based on some surveys, I think last conducted in 2011, at our current rate of phosphate rock consumption, on a global scale, we have about 50 to 100 years left of phosphate rock. And because this is a non-renewable resource, that is all we will have for the foreseeable future through our lifetimes and the lifetimes of our children and grandchildren. And so in that way, phosphate, like petroleum, faces great limitations in terms of scarcity of this non-renewable resource. Like petroleum, where this phosphorus is located is also quite problematic. Of the remaining phosphate reserves, 85% is within 3 countries. That would be Morocco, Algeria, and China. By comparison, and for sort of a litmus test of the potential political concerns associated with having a crucial resource in the hands of a few, oil reserves, about 80% of remaining oil reserves are in the hands of 12 countries. And so potentially issues surrounding petroleum will be mirrored within phosphorus. Nitrogen also faces similar limitations in terms of availability. All, if not most, of the nitrogen fertilizer that we get comes from the Haber-Bosch process. It's a high-temperature, high-pressure thermochemical reaction whereby we take nitrogen from the air, N2, combine it with water as a sort of a hydrogen source in part, and we get out ammonia as a fertilizer. And this system is quite efficient. It's one of the most efficient chemical reactions that we use industrially. But a downside of it, how we currently produce this fertilizer, is that the entire process is powered by natural gas, creating both a demand for more extraction and mining of natural gas reserves, as well as, of course, the natural consequences that this process also involves a proportional amount of CO2. And so this Haber-Bosch process, though it is efficient and though it is able to produce this crucial nutrient, has tremendous impacts on both energy consumption as well as its greenhouse gas emission potential. In addition to the potential issues of running out of these fertilizers, I'll turn our attention back to this issue of our inefficiency. If you take perhaps as 1960 as a measure of our maximum efficiency, that all the nitrogen or phosphorus that we add to the soil is uptaken by these plants, it means that this shaded region in blue, integrated over time, represents all the nitrogen phosphorus that is added to the soil but not uptaken by these plants. This wasted fertilizer kind of comes into play as sort of an economic loss, but there are sort of grander challenges associated with this wasted fertilizer in which we add it to the soil, the plants don't take it up. Where do all these excess nutrients go? The answer is all the roads lead to not Rome, but the lakes, oceans, and rivers. And this is perhaps a sort of an older story within environmental science, that we add these fertilizers to the soil, the plants don't utilize them, and they're carried off by the agricultural runoff into rivers and other bodies of water. Once they're there, the same nutrients that feed plants also feed the algae and cyanobacteria that live in these bodies of water, causing them population to boom and them to grow in number. You often see this as sort of these sort of scummy green overlayers of cyanobacteria in places in which this agricultural runoff has increased their population. Due to sort of normal competition you'd expect for an increasing population of a single species, biodiversity begins to suffer as these algae and cyanobacteria outcompete sort of macrofauna, fish, other things we would like to keep around. But once the algae are able to outcompete these other organisms, they will eventually die off, and during their decomposition, they will release CO2 and methane and a number of other sort of nasty gases, all adding to concerns around this process of eutrophication. So let's say perhaps this crowd isn't the given audience, but perhaps you're not concerned about environmental consequences and inefficiencies. There's still the question of, are our current practices sufficient going forward into the next 50 to 100 years? Many conservative estimates from a number of different groups have placed the global food demand for 2050 to be about twice our current production capacity, meaning within the next 30 years or so, we will need to double the output of every farm on Earth in order to feed the population we expect by 2050. Currently, through all our advances in genetic engineering, improved fertilization schemes, and other sort of traditional tools of agricultural science, we are currently projected to reach about half of this goal, seeing only a 50% increase in global food yields. Now, this creates quite a concern, or quite a practical concern, in which even with our best tricks in our toolbox and ignoring concerns associated with fertilizer use, we're still not on track to meet these food goals and will likely face issues of food shortage within the next few decades. And so the answer to the first question is that, chemical fertilizers work. They work wonderfully. They make plants grow bigger, but they are not without their challenges. And one issue is both damage to the environment, but also issues of availability and perhaps also their efficacy and their ability to feed us going to the next 30 to 40 years. Rather than look at a purely chemical approach to fertilizing plants, modern agricultural science has begun to ask questions about the biological effect of fertilizing plants, caring not about soil chemistry, but soil biology, and in this case, microbiology. So it's quite clear that our business-as-usual model isn't working, isn't sufficient. So I'll have us go back to sort of the drawing board of our picture of agriculture. So within this scheme in which we add these exogenously high amounts of nitrogen and phosphorus to mitigate concerns of environmental damage, the question is, can we forego these external fertilizer inputs? This, of course, places us back in sort of a pre-industrial regime in which we rely upon the natural cycling of our waste back into the agricultural fields. This, of course, is also the model of sustainable and of organic farming, in which you don't rely on external inputs, but rather you try to cycle these nutrients more effectively. A number of years ago, there was sort of a longitudinal 10-year study of conventional versus organic farms in Northern Europe. And what they found over this 10-year study is that the organically managed farms, compared with conventionally managed counterparts that use synthetic fertilizers, are able to produce anywhere between 60 to 90% of the yield as conventionally managed farms. Now, if you're one of these 90% yielding farms, it's great news because you produce basically the same amount of food without adding these extra fertilizers. However, if you're one of these 60% yielding farms, it's quite clear that meeting the food goals of 2050 are not going to be achieved by producing half the amount of food on the same amount of land. For the scientists amongst the group, the natural question is, why is there such a huge variability between these farms? They all follow the same organic farming principles, but why are some 60% yielding and some 90% yielding? The answer, perhaps unsurprisingly, boils down to one of soil microbiology, that the soil is not simply a repository and a sponge for these nutrients, but rather it's a community of living, breathing, and hungry soil bacteria and fungi. This collection of microorganisms we collectively call the soil microbiome are crucial for a number of plant beneficial functions. The insight from the last decade or so has been that the composition of this microbiome, who's living there and what they do, greatly affects what effects you see. Whereby, if you change the composition of your soil microbiome, you change the population distribution, you can increase the performance of these plants' beneficial functions, ultimately leading to higher yields, perhaps pushing you closer to these 90% yielding farms. This idea that the composition of the bacterial community, this microbiome, is important has sort of come at a timely point in science in which the idea of microbiome transplants has become popularized not in just agricultural science, but actually primarily in health sciences. The idea of microbiome transplants may be more familiar from gut microbiome transplants. There's a particularly nasty infection in which your gut, your intestinal system, becomes infected with Clostridium difficile. It's a very, very nasty bacterium that does a lot of unpleasant things to you. And there are currently no sort of antibiotic pharmaceutical options for removing this pathogenic bacterium. Currently, the only way to treat this condition is to introduce a different microbiome to your gut. Usually what happens is that they find an individual with a healthy gut microbiome that doesn't have a C. diff infection. They have a little sample of the feces that they process, and they use that to repopulate your diseased gut with a different population of bacteria. And simply by this different microbiome outcompeting the original bacteria and establishing the correct population, you can treat this infection through this idea of a microbiome transplant. People have sort of taken this idea a bit further, and I'm unsure if it's a good idea to look at things like the skin microbiome. L'Oreal is trying to create cosmetics based around the idea of changing your skin microbiome to get rid of pimples and wrinkles. But for our purposes, the powerful idea is that you can apply this same concept to agriculture and to soil science. You can take an unhealthy soil that has potentially the wrong type of soil bacteria and fungi that produces small plants, here shown in that picture to the top right. And you can find a donor soil that exhibits healthy plant growth, and that would imply a healthy soil microbiome. And simply by sampling a small portion of the soil and using it to repopulate your unhealthy soil, you can now change the composition of the microbiome and improve plant growth simply by adding the right type of bacteria, and you will begin to recover sort of the biological activity of these soils and the plant growth. The question that has sort of now driven the sort of engineering design side of microbiome transplants is, can we actually write a prescription for different soils to treat different conditions and be a little more precise in how we make these transplants? In order to do so, the work requires us to ask three crucial questions. Who's living in this soil microbiome? What are they doing there? What is their function? And most crucially, and what I will talk about from my research, is what do they need? What do we have to feed them to ensure that these bacteria and fungi do what they do best? In terms of what they do, one example are diazotrophs. They are bacteria and fungi that, as their name implies, they eat nitrogen. They are able to do biological nitrogen fixation to take N2, 80% of the air, and convert that to ammonia as a plant-available form of nitrogen. This is sort of the same reaction that we do industrially in the Haber-Bosch process, but now this occurs all in a biological manner at all ambient pressures and temperatures in a much more cheap route. An additional fact about these diazotrophs is that they're able to more readily mediate the availability of these nutrients such that they will never really over-fertilize the soil. And as a result of their ability to sort of hold onto the nitrogen more effectively, you usually mitigate concerns of eutrophication and that these nutrients will never be high enough to leach over and bleed into sort of surrounding bodies of water. Another big place that the soil microbiome provides plant benefits is in terms of inhibiting pathogenic bacteria. There's a strain of Pseudomonas, NPS 3121, that this group studied that normally infects the root systems of plants, leading to a number of different plant disease conditions. What they found is that if you have a certain species of bacillus, this bacterium will move in and recolonize the root system and prevent the pathogenic Pseudomonas species from infecting the root system of, in this case, Arabidopsis, offering sort of a more natural, biologically derived way to prevent plant pathogens rather than using pesticides and other chemical sort of antibiotic components. The perhaps most surprising way that plants receive benefits from the soil microbiome is in terms of environmental resistances. Within the last 10 years or so, microbiologists have sampled the soils in areas that exhibit some extreme conditions, near geothermal hot springs where the soil is very, very hot, around coastal regions where the soil is very, very salty, or in sort of agricultural fields where there's a lot of plant-related disease surrounding them. These microbiologists have found that the resistance to these environmental stressors not necessarily comes from the plant's own genetic code, but rather from the bacteria living commensally or symbiotically with these plant systems. And in the same way that we do microbiome transplants, you can sample some of the soils from these extreme environments, transfer them to a new plant system that has never seen these environmental stressors, and confer things like salt tolerance, drought tolerance, and heat tolerance simply by adding in this microbiological component to help these plants resist these environmental stressors. And so the second question that we've begun to answer is the soil microbiology is quite important for plant growth in that it confers a number of plant beneficial functions, ones that we potentially are unable to recapitulate in sort of chemical methods or genetic engineering methods, and one that requires further study and certainly exploitation to utilize their benefits more effectively. So with recognition that soil biology is quite important for providing these plant beneficial functions, the question is, how should we be farming in the modern age, and how should we be taking care of the soil microbiome? One thing we should stop doing is messing them up. One practice that tends to mess up the soil microbiome is tillage, the process of digging up and loosening the soil to make planting and harvesting a little more simple. However, as you loosen the soil, you start to introduce oxygen deep into the soil and start to expose these formerly anaerobic environments where these bacteria are happily thriving. You start to expose them to oxygen, creating these aerobic environments. And by introducing a different growth condition, you start to change the composition of these soil bacteria. And here you can see both an oxygen plot of a plot of the soil depth versus oxygen concentration, in which very quickly you generate an anaerobic region which favors a certain type of bacteria. And you can even see this genomically as you go deeper into the soil, the sort of genetic makeup of the bacteria living there shifts dramatically. In addition to sort of normal practices of tillage, sort of the general story of the American Midwest has been dramatic changes in the soil microbiome by sort of doing unnatural things. One aspect of sort of plant monoculture that favors certain types of bacteria has been to sort of dramatically shift the composition of the soil microbiomes in these regions. Here you have sort of principal component analyses of both cultivated soil that has been subjected to industrial agriculture, and sort of the native microbiome that is historically there in sort of untouched preserved lands. You also see in these regions sort of a dramatic drop in both taxonomic diversity, the different types of bacteria living there, but quite importantly, the functional diversity, such that in these sort of bluish regions, you're not only changing who's living there, but you're changing what these soil microbiomes are able to do to help these plants grow. And this is perhaps the reason why we've had to rely so heavily on chemical fertilizers due to the loss of these soil microbiomes. So that's what we shouldn't do, but it hasn't answered the question of what we should be doing. And one big thing we must begin to do better is to better feed this soil microbiome. The question, of course, is what do these fungi and bacteria eat? Punchline is that they eat soil organic carbon. Now, these bacteria, they are not photosynthetic, so they can't derive their carbon and energy from CO2 as the plants do. Rather, they are reliant wholly on the residual carbon waste, the agricultural residues, and other carbonaceous species that we normally return to the soils. This usually takes the form of the leaves and the twigs and sort of the other residue that we do not normally eat and consume ourselves. The importance of this residual carbon waste and this sole food source food source for this microbiome has begun to become threatened by certain current practices. One thing that we have begun to do, and again, I think chemists are in part to blame for this practice, is the idea that we can take this carbon waste, all the leaves and twigs that seemingly had no purpose, and we can now use them for something more useful. One practice has been to take these agricultural residue forms and to do biorefining to turn the leaves and twigs into sugars that we can ferment and produce things like biofuels. And while this is certainly a boon for alternative energy and an alternative to petrochemical fuel sources, potentially unforeseen consequence of this is that by converting the sole food source of the soil microbiome to biofuels, which these bacteria cannot use, you begin to starve this soil microbiome, decreasing their population, increasing their biodiversity. And as you destroy the soil microbiome, in the absence of additional fertilizers, quite expectedly, you get smaller plants. And if you have smaller plants, you fix less CO2, remove it from the atmosphere, and you produce less food. This has sort of been borne out by not simply this sort of hypothesis-driven idea, as by taking away the food source of the soil microbiome, you starve the soil microbes, get smaller plants, and fix less CO2. In fact, modeling has begun to show that by removing the food source of the bacteria, making smaller plants, and fixing less CO2, you're actually fixing less CO2 from the atmosphere on a global scale. And by doing a balance of the carbon fixation rates with and without residue removal, modeling has been able to show that by producing biofuels from agricultural waste, taking away from the soil bacteria, the net greenhouse gas emissions of producing this biofuel are roughly the same as burning an equivalent amount of gasoline. And so this process of converting this carbon waste to biofuels is not simply an issue of alternative energy, but now has begun to weigh in on issues surrounding sustainable agriculture. And so, you know, as a chemist, I'm quite loath to say we should stop trying to produce alternative energy and alternative fuels, but it still doesn't solve the problem that this practice has created. And so the work that I have been engaged in, and I'll sort of tell you now for the last bit of the talk, is how do we similarly satisfy needs surrounding alternative energy, but also try to have better stewardship of the soil microbiome by potentially finding an alternative food source for these soil microbes to sort of offset our attempts to turn into biofuels. One practice that has begun to gain popularity is the idea of biochar. In this process of biochar production, it is arguably an ancient one by which we normally would produce charcoal. You sort of take the agricultural residue that you would otherwise convert to biofuel, but instead of sort of burning it and converting it to CO2 at the end of the day, you do a pyrolysis in a low oxygen atmosphere. And so instead of converting this waste to CO2, you convert it to charcoal, elemental carbon. This biochar, this sort of carbonaceous bio-derived mass, you can then actually add back to the soils to continue to add carbon to the soils as a food source for these bacteria, and simultaneously you are able to produce energy as you're still burning this biomass. And it potentially offers a way to satisfy best of both worlds by both providing an alternative energy source, but not depriving the soil microbiome of their sole food source. The work that I have been conducting for the last few years over at Harvard has sort of taken a different approach that actually tries to grapple with the microbes in the soil on a more intimate level. Our work has largely focused on this bacterium called Xanthobacter autotrophicus. It's a naturally occurring root-associated bacterium. You usually find in sort of the root systems around rice fields and other sort of anaerobic environments. Quite curiously, and quite specially, it's also an autotroph, meaning that it grows on hydrogen and is able to use this energy source as a means to fix carbon dioxide from the atmosphere, able to derive all of its carbon for cell growth from this sort of waste product in a process reasonably similar to plants doing photosynthesis. The carbon that it derives from CO2 is used to make biomass and all these sorts of things, but a large portion of it, about 80%, is converted into a polymer that the cell stores inside of its body. This polymer, called polyhydroxybutyrate, PHB, people have usually used as sort of a bioplastic to make plastic cups and water bottles. But the biological purpose of this carbon storage polymer has been to act as sort of a fat reserve for this bacterium. As it's growing under CO2, it will sort of store up PHB. And you can kind of think of it as sort of a bear fattening up in the fall to survive a long winter. This bacteria basically stores an onboard fat reserve as an energy source should it encounter lean times. And so this bacterium is not sort of reliant on soil organic carbon that's added through whatever farming practice, but it sort of brings its own energy and own food source to the party, potentially sort of mitigating the concerns on soil organic carbon. It is itself a form of soil organic carbon, as the PHB stored in this bacterium can be also consumed by other bacteria found within the soil. So this bacterium has an interesting mechanism of converting CO2 from the atmosphere into a form of carbon that it, as a bacterium, and other members of the soil microbiome can consume. Additionally, it's a diazotroph, as I mentioned before. It converts atmospheric nitrogen into ammonia via the enzyme nitrogenase. And it takes this nitrogen and stores it in its cell body, sort of acting as a delivery vehicle for this plant nutrient. And any sort of phosphorus it encounters while it's growing, it stores as these polyphosphate granules in its cell body. We've become quite interested in this bacterium as it is sort of an omnibus of plant fertilizing action, where it carries on, it brings to the soil a source of carbon derived from CO2 that can feed itself and other bacteria, as well as it's able to do nitrogen cycling and also accumulate and mediate phosphorus release. My work has sort of always been as sort of the interface between chemistry and biology, trying to find ways that we as chemists can sort of dial back the damage that we did historically. And our goal has always been to find ways to link an alternative form of energy, in this case something like renewable electricity, to generate products for sustainable agriculture, in this case potentially a bacterial-based soil amendment. In our view, this naturally occurring nitrogen fixation cycle, in which you have bacteria in your soil microbiome that fix atmospheric nitrogen into ammonia. This is still powered by naturally occurring and naturally returned agricultural waste, but as we talked about throughout this talk, this process is still fairly limited and fairly bottlenecked by our ability to add as much of this energy source, this carbon waste, as we can. Our approach seeks to sort of augment this process by not relying upon the natural energy flow through the system, but sort of supercharging this process with renewable alternative energy. In our scheme, we think that if we can grow these types of Xanthobacter omnibus fertilizing bacteria with an alternative energy source, we can accumulate it and add it to the soil to increase the flux of carbon and nitrogen through the natural soil microbiome. In order to culture these bacteria, we've relied upon renewable electricity in sort of an electrochemical mode, sort of blending the chemistry with biology. In this sort of bioelectrochemical reactor, we have a pair of catalysts and a pair of electrodes that are able to split water into oxygen, as well as a food source for this bacterium. This bacterium growing here can take nitrogen and CO2 from the atmosphere to cycle these both through back to the soils with an additional energy source derived from this electricity, as well as a higher flux of these crucial nutrients. How this works mechanistically is that at our cathode, we're able to split water or protons and combine them as hydrogen gas, which feeds the bacterium through the naturally occurring hydrogenases. This energy source and electron source is used both to fix CO2 into biomass in the PHB, this sort of fat reserve that this bacterium brings along, as well as drive the fixation of nitrogen to ammonia, bringing nitrogen back to the soil from the atmosphere. Now, so this is sort of the chemical side and electrochemical side of it, and whenever you do research, you should always try to think as far down the process as you can and try to test how good the things you make are, to actually validate if they're designed or if they're designed properly. So to actually test whether these bacteria that we can grow electrochemically actually act as a beneficial plant fertilizer, I sort of did things that chemists don't normally do. I did some greenhouse work. Was conducted down at the Arnold Arboretum down by Jamaica Plain. In their greenhouses there, and I picked radishes as a model crop because they grow super quick, about 3 weeks from seed to harvest. I basically tested whether the bacteria that we can produce through this alternative route are able to increase plant growth. And so our initial forays have shown that by adding more of this Danthabacter-based bacterium, we get bigger radishes, basically. And for the amount of bacteria that we've added in these experiments, we get a roughly 2-fold increase in the yield of these radishes through a biofertilizing mechanism that we're potentially still teasing apart. But we imagine that it is very crucially dependent on this bacterium's ability to carry carbon to the soil, as well as do nitrogen cycling through nitrogen fixation. This, of course, is still sort of work in its early days of progress. And one of the big things that we're still puzzling over is how our unusual addition of this Danthabacter is able to interact with the native soil microbiome in ways that we're still exploring. So far, we've shown that this bacterium is not harmful in any way to the soil, that if we add this bacterium to the native soil microbiome, we really see no dramatic change, as the native microbiome is probably as good as this radish needs. But if we have sort of sterilized soil where the biological activity has been lost, potentially simulating what you see across a lot of farms in the American Midwest, addition of this bacterial fertilizer can sort of recover the growth of this soil by replacing this biological activity that had been lost. Of course, we're also beginning to now compare it also with other types of bacteria that you may add to the soil, things like Bradyrhizobium japonicum, which is a symbiotic nitrogen fixer, which is not evolved to work well with radishes, so it's unsurprising that this very specific bacteria doesn't act as a good fertilizer. But Danthabacter is more of a generalist. It doesn't really care what type of plant it grows near. It still releases ammonia, and potentially that's why we see an improved effect of our Danthabacter grown electrochemically than other types of soil amendments. And so for the issues of what should we be doing to actually benefit these soils and to better take stewardship of these soil microbiomes, it's still an evolving challenge from the technological and, of course, the scientific study end. But there are some key insights that we have, most of them centered around the issues of making sure that this active, living, healthy community of bacteria has a food source, and that we are not depriving it of its crucial soil organic carbon. And potentially there are technological ways that we can sort of augment this cycle by finding new ways to add additional carbon, potentially derived from things like renewable electricity. And so I'll start to wrap up, and sort of the takeaways that I'll leave you from this talk are sort of threefold. First is that our current use of synthetic fertilizers is very effective, but it's likely unsustainable and will probably not be sufficient going to the next 30 to 50 years. Rather, we should start thinking of the soil as not a chemical entity, but a biological entity, and to appreciate for both its benefits, but also its requirements as a living, breathing, and hungry entity. And in order to do so, one of the most crucial things we must start to think about is not simply who's living there within the soil, but how they are fed and how they derive their energy to do all their wonderful biological activity. And so with that, I think I will sort of end there. And of course, I'd like to thank you for your attention and this invitation to tell you a bit about what a chemist thinks about soil ecology. I'd be happy to take any questions should they have arisen. So thank you.
Thanks, that was really interesting. I'm just curious more about this Danthabacter. So it sounds like it's an anaerobe, yeah?
It's flexible.
It's flexible, okay.
So certainly the nitrogen fixation is best done in a microaerophilic environment.
Okay.
And so having, I had a little bit about the tillage before, and that once you establish your slightly anaerobic environment beneath the soil, it's very crucial not to upset that and to introduce oxygen, or else these guys as well do not fix nitrogen.
I see. So I guess I was going to ask about, so in this example, you're sort of growing them in the lab, you know, with electrodes, providing them food, and then you're applying them kind of as a fertilizer.
Yeah.
But I'm thinking about rice, which is a crop that grows in more anaerobic conditions and it's a crop that feeds a huge amount of people. So I'm curious if this is something that could be used in rice by just sort of introducing the bacteria and letting them do their thing.
Yeah, so certainly one key issue still is how do you continually add a food source for these systems. And so you would still require, so once Xanthobacter runs out of PHB, they'll start to die and they won't be functional anymore. So one idea has been that, currently we have the bioelectrochemical reactor separated away from the farm, but we've been thinking about sort of integrated methods in which, for something like a rice paddy in which it grows in water, and you could use it as an electrochemical reactor, could you sort of have an integrated system sort of built with producing the bacteria in place within the agricultural context? It's still sort of a far-fetched idea, as now you have to start sticking electrodes all across your rice paddy. But as precision agriculture becomes a thing and people are sort of digitizing, monitoring soils, it's a possibility as well.
Thank you, Kelsey. This was really fascinating. The more I learn about microbes, the more blown away I am.
They do a whole lot of things.
And so I had a couple of questions. The first one is, it seems that they are very self-sufficient. I'm talking about your bacteria. What do they get from the plant? And the second question is, have you seen a limit to how much they can increase the yield of the plants?
Right. So in terms of what their actual relationship to the plant is, these bacteria haven't been as widely studied from the pure plant-microbe interaction standpoint as, B. japonicum has. And so for the limited knowledge that we have is that there is still some organic acids that the plant will secrete to sort of mildly feed these bacteria. And so there is potentially a symbiotic relationship there, but it hasn't been as well characterized. In terms of sort of the limits to how effective this type of fertilization plan could be, we really haven't been able to push it to the far extreme, as ultimately, we are limited to a laboratory-sized sort of bioreactor. And ultimately, sort of the goal is to scale this up and to do field studies in sort of larger agricultural contexts, which requires a ton of bacteria to grow. So that's sort of out there, but we haven't sort of hit a limit yet in how much they are able to fertilize.
Thank you for a wonderful presentation. One of the things that really strikes me is how wonderfully interdisciplinary this presentation is. And one of my questions for you is if students were inspired, I teach the Lunch and Learn seminar that's based on this, and one of my questions is, students are inspired for their careers by this talk. What are some of the range of training, background, disciplines? Who's part of your team? What would you recommend students with different pieces of interest in different pieces of this puzzle consider for future career paths?
Right. So it is terribly interdisciplinary, and I have come into this thing as a chemist who has had to very quickly learn biology, and that is certainly a feasible route. I've done it. It requires a bit of checking some of your assumptions and how easy biology might be. It's fairly difficult. But certainly these sort of principles have come from, or these sort of works have come from, establishing a solid understanding of one aspect of these sort of interdisciplinary things. So me as a chemist, or perhaps someone as an engineer or bioengineer. And it does, at this point, it does rely upon finding good collaborators who are able to bring the other half of the interdisciplinary work to your thing. So for this sort of work, we are now working very broadly with people within systems biology who understand the microbe-plant interactions a little better than I as a chemist can do. But also a range of engineering disciplines to apply sort of the quantitative understanding of mass transfer in and out of the soil system, and certainly also energy transfer in and out of the soil system. And so this is sort of a nice work that has really touched on most disciplines, and sort of everything can bring an aspect to it. Even things within the social sciences, looking at sort of economic considerations of these things, actual practice, and how do you get people to use the system, are all sort of beginning to weigh in on this. It is a very interdisciplinary pursuit.
I'd like to also thank you. So one of the things that's interesting in using radish is that the plants in that family don't have mycorrhizal associations, which are so critical to the phosphorus cycle.
Right.
And so my question have you tried it on different plant species and look at it in association with other microbial symbionts that can be so important to plant growth and nutrition?
Right. And so, you know, radishes were picked because I'm very impatient. And experiments longer than 3 weeks are sort of unthinkable for a chemist. But certainly one of the big drawbacks of picking radishes is that they are fairly simple, they're fast-growing, and don't exhibit a lot of the more complex plant biology that things like corn or soybeans would. And so it's not something we've been able to do so far within our limited abilities, but it's a much more intriguing application in that one of the big advantages of biological fertilizers over chemical ones is that they're much longer lived. They are slow release, they're able to persist much longer in the season. Over the course of 6 months of growing corn, you may only need one addition of your biofertilizer rather than weekly additions of a chemical fertilizer. So that's certainly one aspect of the actual full potential of this biological approach that we haven't explored, but that actually dealing with sort of longer lived or longer growing plants is sort of the next step that we have to take this.
Thanks for this. So in my experience with bacteria in general, I know that phages are a really big issue working with bacteria in the industry. How susceptible is X. autotrophicus to phages, and how have you guys kind of dealt with that issue?
That's something that we haven't really dived too deeply into. So we're unsure. As we've been adding it, we have added perhaps unreasonable amount of this bacterium to the soil. So we calculated that the amount of bacteria that we add to see some of the highest effects is about 1,000 more in population than the naturally occurring prevalence of Xanthobacter autotrophicus. And so certainly having such a high population of these bacteria potentially sort of buffers out issues with phages and other sorts of actual interaction with these real-world settings. So, as chemists, we haven't been able to dive as deeply into these very important biological considerations. But if there are people who are interested, we'd be very happy to collaborate and look at these things.
Well, thank you again once more. Let's give him a big hand.
Thank you.
I'm sure he'd be willing to talk to people up in the front for a bit if you're interested. And once again, hope you'll come back for another